device to monitor maturation of engineered tissues in a bioreactor. I'm Daisy, this is Denise, that is Julie, and this is Andrew. Our outline, we're going to go over the background and motivation of the design. We're going to give you information about how the bioreactor and the OCT work, the expected results, our design and the construction of it, then the device and system performance, and finally our concluding statement. So for the background of our design is currently scaffolds are used to grow tissue, but this tissue has to be maintained in certain sterile conditions with a constant temperature and a contaminant-free environment. And with our design, we're working with a bioreactor, and the bioreactor is used to compress and decompress cartilage tissue in order to stimulate its growth.
The motivation behind our device is, as of right now, it's currently very inefficient to work with bioreactors in the sense that every time you want to grow a batch of cells of engineered tissue within the bioreactor inside of the well plates, you really don't know where you're at in the development of your cells, because every time you want to observe how they're maturing in development and growth, you have to actually take them out of their sterile environment that Denise was talking about. The bioreactor requires the conditions to be very sterile, and as of right now, there are no current methods to actually monitor those cells inside of the bioreactor without actually taking them out and rendering them useless due to contamination. So that's what our RAM is trying to, that's kind of what inspired this idea to go ahead and move forward with actually trying to observe the development of these cells within the bioreactor instead of having to take them out every time you want to see how your cells are developing. This is an example of the bioreactor that Dr. Nam and his lab are working on, and they have engineered and designed. So what a bioreactor does is, this one's specifically for cartilage, and right there where it says like culture media, those are the. Yeah, please, thank you. So these right here, these are the wells where the cartilage are situated in. All 24 wells. Yeah, there are 24 wells inside of the bioreactor with cartilage inside each and every one of them. So what the bioreactor is intended to do is to stimulate the cartilage by compressing and decompressing on the engineered tissue. So this would stimulate the growth of cells. And if I actually had an image, I would show you where we would be trying to replace all the RAMs so that you could actually shoot a laser through the RAM, a hollow version of the RAM, and be able to image what is developing inside the well plates. But yeah, again, so in order to prevent contamination of these cells, to monitor them and to actually see if any growth is happening on the scaffolds, we want to create, our idea was to create a RAM focused so that with a series of lasers and mirrors and lenses that would allow us to observe the growth within the bioreactor. Okay. So just to explain the problem again, having to take out your cells every time you want to monitor the growth on the scaffold is very time consuming. Imagine having to grow it and then take it out and then start over again just to see how it's developing. Also, cells can get very costly, so this would in essence, be saving time and money. So.
It's starting up now.
So is the ramp exercising the cells?
Yes, it's still stimulating cartilage because we were able to, in the later slides we'll show you the actual breakdown of everything that's being incorporated into the RAM. But there's a layer of glass underneath where we hollowed it out to actually press against the cells as it's compressing and decompressing.
There's a moving platform that raises the cell culture to hit against the RAM, and then the RAM causes compression.
How do you know it's in contact?
Well, just by judging with the eye, I mean, we can see that it's actually compressing and decompressing. We have an image of it actually in our slides, and it will show more up close picture of the contact.
You're contacting the cells, right?
Yes.
And when you say you can see it, you're saying you see it through a microscope?
Through a microscope, not exactly. We haven't tested that part. Do you want to go to the demonstration? We'll just do demonstration. Go ahead. So as I was explaining about the bioreactor, here's a basic diagram of what the bioreactor that Dr. Non's lab has created.
Okay, so OCT is optical imaging tomography, which is very similar to optical ultrasound imaging. The similarity is that they both measure the time of reflection of kind of like a reflected echo. OCT uses low coherence interferometer rather than reflection time measurement, and it has relatively long wavelength flight, so we can penetrate more into the medium. On the figure shown on the left, the OCT setup, the laser will be shined from the laser source, and it will hit the lateral scan, which will rotate around to move the direction of X and Y direction, and it will go into the sample. And then when it hits the sample, it will reflect the laser beam light, which will hit the lateral scan again, and then it will be split from here, the beam splitter, and the reflected laser will go into the detector, and we'll see the image.
So again, the objective of the device design is to construct a RAM which will allow us to visualize what's inside the bioreactor themselves and integrate OCT imaging that's what we're going to be using to view the images. So this is the device setup. As you can see, this is the same bioreactor, except for we're going to have a modified RAM, which is then light is coming in through the OCT optical fiber, goes through a collimator to make sure all the light is parallel, and then goes into the galvo system. The galvo system will be used to scan so that we can have an entire scan of the scaffold and then go into our modified RAM. This is the design of the RAM. As you can see, we have a side window in order to allow for the laser to be going through. Or the figure, part C of the figure shows the top portion intact of the RAM, which would have a reflective surface, and this is going to guide the light down through the RAM. Lens going to be placed on the side, another lens going to be placed in the middle, and then we have a transparent glass window that's going to be at the bottom, and that's going to be in contact with the scaffolds themselves. Here is the other part of the RAM design. When we were joining it with the OCT system, we needed a third lens, and this was so that the collimated light could go through this lens, focus on the second lens, and then go down through it and refocus again on the engineered tissue. And as you can see, figure B is our actual assembled RAM with the lenses all in place, and the metal holder for the lens. In any case where you would want to change maybe the distance between the entire system, because we're working on a really small scale right now, the third lens is able to be adjusted, so you can replace that with a different focal length in order to get everything focused properly.
So as you can see on this one is a bioreactor that I was talking about, and that's the feeder of OCT setup that we have. And this one is the galvo that has 2 mirrors right there, and it will rotate around to see the different directions. Since the controller of the galvo machine is not working, so we just measuring the power which is coming out from the RAM, from the laser. So as you can see, the power of the laser beam is going up when I focus it onto the RAM.
What Julie was showing you here just now was, so right now there's virtually 0 beams right here. But as we move closer to the actual laser beam, we actually see that it increases by 250. If we have it perfectly aligned, we can get the power meter to actually read closer to 350, which would be a lot better for our imaging purposes. That's at the very top, the 1.78. That is what's originally coming out. That's the direct reading from just this machine alone. But after we introduce the RAM to the system, we see that it is decreased to 350. So there are still, there's a lot of energy not being, that's being dissipated throughout the RAM. That's one of the issues that we need to address and build upon.
So in conclusion, our system, the OCT system used in conjunction with our RAM design did prove successful. We did obtain a functioning system. The only thing was we couldn't obtain power when we used the galvo, just only when we put the RAM right next to the collimator. This design is much needed for tissue engineering, so I'm sure the idea will catch on quickly. For future work, we do have some things to work on. Currently we only have one RAM. So because we're in a 4x6 wool plate, we will try to build 6 RAMs and then work from there. So this way they can image much more tissues at a faster time and output results at a faster rate. We're also trying to image from the bioreactor, see if, while it's compressing, see if the imaging is working properly. Right now, we polished the cap in order to make it reflective, but we only obtained 78% reflectivity, which I'm sure that is affecting the power rating. So in future, we would like to see if we couldn't do more with the cap, so maybe use an actual mirror and then obtain a power rating for the whole system, including the galvo. We'd like to acknowledge Dr. Nam and his grad student Brandon, and Dr. Park and his grad student Dollar and Charlie.
About 30 seconds for questions. So these alternative unfocal microscopy.
OCT works with this because the way it sends the laser to and then collects back the readings. But I mean, you could redesign maybe a system so that you could use a different imaging modality.
Of course, we need to make improvements such as making the system or the RAM a lot more energy efficient, I guess, so that a lot of the images is created throughout the RAM because that only getting 30% is not that great. We would like it more to be an 80%, but with the little improvements on the computer.
Quick question. How do you know the OCT make a charge the material mature? Why is biomaterial mature?
You see changes in the scaffold itself when you image it, and that's what we're assuming to be the growth of the tissue, the development of it, the differences that you see in periodically.
So this is one type of image tool.
Yeah.
Imaging the scaffold changes, right?
Yeah.
Not internally the cell maturation.
You see the changes in the scaffold structure. That's what we're taking to be the growth of the tissue. But I mean, yes, you would have to. It'd be best to couple it with a different imaging to make sure that you actually do have cell growth. So that would be a better way to test to make sure that our system gives us that useful information that is relative to what we're trying to do.
Does the fact that one of the gobble axes is that why the percentage is so low?
When we were using the Java, one of the axes wasn't working properly, so it wasn't putting the light directly through it, and we think that was probably why we couldn't get a reading. No?
Doesn't matter at all.
Okay. So, I mean, we think is when we aligned it, like, it just was not going through properly.
Let's move to the next group.